right, good morning, everyone. I am Glynnis Gary, and I'm here with Dr. Christopher Toepfer, who is the recipient of the BCVS Outstanding Early Career Investigator Award. Christopher is a Sir Henry Dale Fellow at the University of Oxford, where he started his laboratory focused on studying the mechanisms and identifying therapies for a form of genetic heart disease called hypertrophic cardiomyopathy. So, Christopher, you gave an outstanding talk yesterday, and on behalf of BCVS and the Early Career Committee, we congratulate you on this huge accomplishment. That's really fantastic.
Thank you very much. It was a really beautiful session in general. All of the other speakers as well did a brilliant job, and it's really a lovely thing to see so much enthusiasm in the session as well.
Yeah, absolutely. Yeah, congratulations to all of the finalists. It was fantastic science that was presented. So let's kick things off by having you share with the audience a bit of the background and findings of your study.
So what I guess we really established that two different major causes of hypertrophic cardiomyopathy are actually very different in the way that they function in the long term. And we should think about maybe treating those diseases as two separate entities and maybe also designing new therapies for some of them too. But it was really quite striking that these two diseases at the organ level that look very similar, at the molecular level are very different. And that's important. And I think that's going to be very important for treating people in the future.
I'm wondering if you could talk a little bit about the development of the assays that were used in this study that really empowered you guys to dissect the variants of these diseases.
So the assays are based around some very simple principles in HCM, which is trying to understand why there's hypercontractility and why there's abnormal and slowed relaxation in the disease. And it was a really lovely, large team effort of computer scientists, us molecular biologists, genome engineers, to bring this platform together to be able to measure this in induced pluripotent stem cell-derived cardiomyocytes. And that really allowed us to gain insight into disease in a human model of the disease, which was really lovely to see. So we don't have to do these experiments in other animal models.
For the audience, you utilized beautifully a drug, mavacamten, that's currently in clinical trials. I'm wondering if you could talk a little bit about that and about the implications for this as a translational study. For me, it was personally a very inspiring story to see of personalized medicine and bench to bedside discovery. So if you could talk a little bit about that, that would be great.
Mavacamten has been incredibly useful at the level for us basic scientists to be able to start to dissect what's really happening in the disease too. So what I think we found is that in a large subset of HCM variants, when we could treat the cells with mavacamten, a lot of the different things that were going wrong inside those cells would go away. And that was very clearly by directly treating the mechanism of disease in those specific subset of patients. So we have to see, I guess, I'm not best placed to talk about the clinical implications long term and whether that'll apply to other populations of people with this disease. But I think there's also further important things to understand in the disease still, and mavacamten has been instrumental in helping us to understand those things.
What are the next steps for this project that really excite you?
So I think there's a lot to do here. What we are really, really excited about at the moment is the idea that we could start to bin different variants that people may have and allow us to then therapeutically treat them in a better way. So how we're doing that is we can't, at this moment in time, go away and generate every single patient line in a dish. What we can do is we can start to use some of this data to feed computational models to make predictions about what might be causing disease in certain patients. And I think that might be the future for us in some ways, that we could start to help and direct treatment for those patients by using a sort of hybrid approach of biology in a dish and also modeling on computers to try and really understand deep mechanism to help people.
It's very inspiring work and beautiful study. I guess in the next 10 years, where do you hope that this space is for hypertrophic cardiomyopathy, and where do you see it going?
I think we'd all love to see that tomorrow we'd have the golden cure for all cardiovascular disease. Unfortunately, I think we've got a little way to go still, but it's very exciting that we're seeing such rapid developments in this field. And there seems to be so much real interest now from biopharmaceutical companies and lots of scientists obviously working together now in tandem to try and understand these diseases in far more depth and understand fundamentally how the heart works. And I think that's really where I draw my inspiration from, is that the heart is a magnificent organ. You're born with it. It beats billions of times in your life, and it functions for 80 plus years and just does that on its own. So I think that's what we're really, really enthusiastic to understand more about and the implications of that going forward.
Yeah, fantastic. I wholeheartedly agree. Well, we thank you, Dr. Toepfer, for your time and your dedication to the BCVS and AHA over the years. I know you've mentored many students, both from Sarnoff and HHMI, and your dedication to promoting scientists and physician scientists has been extraordinary. So on behalf of the BCVS, we congratulate you on the Outstanding Early Career Investigator Award and on your phenomenal science. We know you'll be a leader in cardiovascular science, and we look forward to your pioneering discoveries to come.
Thank you very much.